everybody. Today I want to walk you through the investigation that we did to see how we can simulate what happens to food molecules in the small intestine. We used a solution of two different types of food molecules. We had starch in water, which is a complex carbohydrate, often found in a lot of the foods that we eat. And we also had glucose in water. Remember, glucose is also a carbohydrate. It's simpler. It is sugar, essentially. So what we wanted to see is if we had glucose and starch in our small intestine, would it potentially be able to move through the walls? Because we're trying to figure out what's happening to these food molecules that seemingly disappear in the small intestine. We don't have a small intestine to use, so we're going to be using a dialysis tube, which has similar properties to the small intestine. Now, what I did is that we took the starch solution and the glucose solution, and we placed them into the dialysis tubing, tied off both ends, and put it in a beaker of water overnight. To make things a little bit simpler for us, I took all of the liquid that was inside a bunch of the dialysis tubes and I poured it into a collective beaker. I labeled it inside. I also took the water that was left over in the beaker and put it in another beaker, saying outside. So I have the liquid that was inside the dialysis tubing overnight, and I have the liquid that ended up in the outside, the water, and if anything made it through, it should be in this liquid. Now, reminder, we're looking for two food molecules. We're looking for glucose, which I can't even tell is in here, and we're looking for starch, which is a little bit easier to see, but in smaller concentrations, it wouldn't make this milky white sort of color. We can't really detect it with our naked eye or rely on that to know if it's present inside the tube or outside. So what are we going to use? We're going to use indicators. Indicators are substances that change color in the presence of other chemicals. Iodine is an indicator for starch. On its own, iodine is kind of like a brownish orangish color, same when you mix it with water, but when it identifies starch, it's going to turn black in color. Benedict solution is an indicator for glucose. It on its own is this bright blue color, but if it detects glucose, it's going to turn an orange color. So in the lab, we set up some microcentrifuge tubes, which are these little tubes that have caps to test the liquid both from inside the tubing and outside the beaker to see what stayed in the tube and to see what made it through. We labeled them with letters. IB would be the inside the tube tested with Benedict's. II would be the inside the tube tested with iodine, and then OB and OI, outside Benedict's and outside iodine. We're going to start with the test for outside. So looking at my tubes, my first one I'm going to do is the outside, there's my IB, outside Benedict's. So I have outside Benedict's, I have inside Benedict's, and this is going to be outside iodine. All right, so my outside Benedict's, I'm going to take some of the outside liquid, put it into the tube, and then I'm going to take about 3 drops of my Benedict's, cap and shake. Now the thing about Benedict's is it only works when it's put into a cup of hot water. So I'm going to have my cup of hot water, I'm going to put it into the side here, and we're going to see what happens. Outside iodine. So we're taking my outside liquid. This is looking for starch because I'm using iodine, and I'm going to put a few drops of iodine indicator into it and see what happens. Doesn't seem like it changed color. So my outside iodine stayed kind of this orangish color, showing that there is no starch outside of the tube. Let's see if anything happened. Ooh, okay. So now I have over here with my hot water, my outside Benedict's, and look, it changed to this orange color, telling me that there is glucose outside of the tube. The Benedict solution reacted to it being outside. All right, so what we see is outside, no starch, iodine did not react. Outside, Benedict's turned orange, tells us that there is glucose outside of the tube. Let's see what happened for the inside liquid. So here's my inside. Again, let's check for our Benedict's first. So I'm going to use my blue Benedict's to look for glucose. It needs to go for a little hot tub. Wait on that. My inside, looking for starch. Is that right? So inside iodine. Ooh, I think I'm seeing something happen here. Ooh, this is changing color. So my inside is pretty much a change for my iodine, saying that there is starch inside the tube. Let's see if anything happened with glucose. So my inside Benedict's also reacted. So it tells me that there is glucose inside the tube. So what do these findings tell us? They tell me that my glucose was able to make it out of the tube because the Benedict's reacted outside of the tube, but my starch could not. Inside the tube, there was still some starch there, which makes sense because it didn't leave, but there's also some glucose there as well, so not everything left. Hopefully this helps you understand this activity better.